Okay. Um, I know that there were at least a couple of you that missed a lot of time last week. Um, so in order to kind of summarize what's been done, I thought I'd do a quick recap of the earlier lessons. Okay, so you're going to have to watch as we go through the different handouts. Okay, and I'll try and summarize as best that I can what we did. Okay, otherwise you can try and ask maybe Miss Ramsey, or you can ask substitute Miss Johnson. She might remember a lot of this stuff too. Um, Miss Rogers may remember some of her genetics because she has biology background, kinesiology. Okay. Um, I'd like to say that I can get on to Dim Dim with you before the test, but um, having some trouble accessing internet. So if I can, then I'll let you know. But hopefully this will help in the meantime. Okay. So um, this chapter is chapter 16 on genetics and heredity. Um, we, I put this up at the first just to kind of get people interested, and these were some of the things that they looked at in one of the little labs. So if you didn't do the lab, you should have a look at the lab, go through it yourself, ask someone who has done theirs to explain things to you, okay, so that you can kind of understand and get a little bit more out of what we had done as some of the activities. Okay, so this was looking at some of the traits that showed dominant and recessive properties. So complete dominance and single traits. Okay, first we started off by looking at some terms that you have to really understand for this chapter. Um, First one is, of course, traits. If you don't understand what a trait is, then it's really difficult to make sense of it when we talk about it all the time. So a trait is basically a distinguishing characteristic. We have a whole lot of them that make us each unique. They are each determined by a specific gene or combination of genes in our chromosomes. Some may be altered by our environment. So for instance, you might be born with a certain hair color, but when you go out in the sun, then the sun will lighten the hair. Or if you go to the hairdresser, then you might change your hair color. So our environment can also affect these traits. The next one is heredity. Heredity is basically the transmission of traits from one generation to another. If a trait has been passed on, it's said to be inherited. Selective breeding, just like the name suggests, it's when um, they kind of force different matings in order to try and get a line or a lineage of organisms that have specific desirable traits. So for instance, they may have certain plants that they want to have resistance to a certain um, bug. So if they notice that certain ones are more resistant, then they'll try and breed so they only get that type. Um, for dogs, maybe they like to have a certain build in the dog. Jillian, I remember, was sharing with us that she had watched a documentary um, where they were dog breeding, and there's certain dogs that I think they were maybe chow chows that she was talking about, and they have the really big head. Um, but because they have the big head and shoulders, they've bred it to the point where um, the mothers can't have normal births. They have to have C-sections. So if you can imagine, dogs having to have C-sections every time. So sometimes it's not the best for the organisms. Like certain other dogs, because of their selective breeding, um, they have problems with their hips or problems with their um, different joints and things like that. So Sometimes it's not the best. Genetics is a branch of biology that deals with principles of variation and inheritance in animals and plants. Um, they use things like um, Punnett squares and pedigree charts and things like that in order to determine the likelihood of inheriting certain traits and predict patterns of inheritance. So we'll look at probabilities, things like that. So that's what we're going to be talking about for this chapter. Bit of history. I said early on they really didn't know how babies were conceived, where they came about. So if you can imagine, way back in the early days, they'd have no idea. Okay, Hippocrates thought that each part of the organism came from certain seeds. So seeds of the parents that fused together to form an individual. That was his thought. In the 1700s, the scientists believed that the male was the only real contributor, that females only carried the baby. So basically, it was like a mini baby was inside of each sperm, and the sperm was implanted into the womb of the woman or the uterus of the woman, and that was her only job, was to carry this baby to term. Imagine. Along came this fella, Gregor Mendel, and you could consider him probably the father of genetics. His studies, of course, like a lot of scientists, were never really found to be all that important or believed until after his death. But he spent 8 years in a monastery. He was a monk studying pea plants. Um, and through his studies, he was able to show a lot of inheritance patterns and prove, because he was a botanist and he was a really good mathematician. He was very particular and conducted precise, controlled experiments, repeated things many, many times. So he had excellent results that eventually, when they were understood better, um, were the foundation for a lot of the genetics that we understand today. So we did watch a little video about him, and there's links to the videos and there's links to different little animations on the blog. So please have a look at those because they will really help you out understand things a little better. Okay, there were two questions that I asked, and then I realized that I actually gave you the answers in the handout. So the first was, why was the common pea plant an excellent choice for his experiments? And second was, what were the 7 characteristics that he looked at? And you should be able to tell me some of the characteristics and the 2 forms of each. Okay, so pea plant was a really good choice for him because they were commercially available, so easy to get, easy to grow, matured quickly. They had a lot of babies quickly. He could use them to self-pollinate and make sure that there was no cross-contamination because the sex organs were entirely closed in the flower. Okay, so that was important so that he didn't have any cross-contamination between things either. He also was able to use different varieties that had different traits, and these could be easily observed through generations. So rather than using a cow, for instance, a cow can only have one baby at a time, and it takes a long time for them to for their gestation periods. Okay, peas were a really good choice because they had um, a lot of traits he could look at. They were easy to get, easy to grow, could get many offspring, so lots and lots of results. The 7 traits that he did look at were seed shape. Okay, so seed shape, if you think of it's a pea plant, so the seeds are actually the peas. Okay, so seed shape was either round or wrinkled. Seed color was yellow or green. Flower color was purple or white. Pod shape is, this one says inflated and constricted, but you could say smooth or wrinkled. Much easier to understand. Pod color was green or yellow. Flower position is axial or terminal. So if you look at these ones, axial is here, let me see if I can get a little pen. So the axial, the flowers are out to the sides, whereas terminal, they're at the end. Terminal is like end. If you think of a bus terminal, it's the end of the stop. The last one is stem height, or you could just say plant height. So either tall or short. So the ones on the left are the dominant traits that you'll find that were dominant. And as he did his studies, he found with single traits and with his dihybrid crosses, he found that, oh, I'll explain it after anyway. Okay, so the video is really good to watch for this part, rather than have me explain it all again. But anyhow, what he did basically was he performed two different general types of experiments. The first type of experiment was called a monohybrid cross. Mono meaning one, so he looked at one trait at a time. He kept all other things constant, so that made things controlled. So if he was looking at pea shape or seed shape, then the flower colors and the plant heights and all the other things were kept the same between the plants, and he only looked at the pea shape. So that meant he had to get purebred plants that had the trait he wanted to study. So by using selective breeding, in other words, once he got plants that had a certain trait, he kept on breeding them until every time he bred them, he got that particular trait. They were true breeding for that trait. He called that the parent generation. Okay, so the two parents that he would breed, he'd have one that was purebred for the dominant trait, okay, which he didn't realize was dominant at the time, but for instance, one that was purebred for tall and the other that was purebred for the other form, so one that was purebred for short. He crossed them, in other words, mated them, took the pollen from one and pollinated the eggs or ovules of the other. Okay, and he looked at the traits in the offspring. So the offspring of the purebreds was called the first filial generation, or F1 generation. Then he took those offspring and he'd let them self-pollinate. In other words, they'd mate with ones that have the exact same genotype. Okay, if they're mating with themselves, then they use the pollen and they fertilize or pollinate the ovules on the same plant. Okay, since it's the same plant, they're going to have the exact same genes or exact same alleles. You're going to understand those terms better after. Okay. When he mated them with themselves and he got the next set of offspring, those set of offspring are called the second filial generation, or F2 generation. Okay, so you should understand what monohybrid means. When he did the monohybrid cross, the parents are the purebreds, so tall and short. Okay, when you're talking about that particular trait for the height of the plant, so tall and short were mated to get the first filial generation. And he looked at those offspring, then he mated them with themselves, and he looked at their offspring, the second generation. So what he found was when he mated, so let's see if I can get you to focus. When he mated the short with the tall, all of the babies in the first filial generation ended up being tall. Okay, so if he took some of these plants, some of the baby plants in the F1 generation and self-pollinated them or mated them with themselves, okay, he ended up getting 3 tall and 1 short. So we went back and we looked at what they must have had in terms of alleles, okay, or in terms of um, the factors that they must have been able to pass on. So we said, okay, well, short seems to be hidden in the F1 generation. You see? Whereas tall seems to show up. It's the only one that's being seen. So we said that must be the dominant trait. And short seems to be the recessive trait. So we used the letters in order to represent. The big letters represent the dominant trait, and the little letters represent the recessive trait. Okay, so if they're purebred for a particular trait, then they must have both of the same alleles or both of the same genes, what we know as genes. Okay, so the mom gene was a little t and the father copy was a little t. Okay, in this particular plant. So in the short plant, it was little t, the 2 recessive genes. And in the tall plant, it's big T, big T. So when these 2 were mated, the only gene that the short plant could give was little t. Okay, so all the baby plants in the F1 generation must have gotten a little t from the short plant parent and a big T from the tall plant parent. Okay, so when they have one of each kind of allele or one of each kind of gene that's possible, then we call them heterozygous. So they're heterozygous and they have one big T, one little t. Each of the parent plants are homozygous. Okay, meaning homozygous means that they have 2 of the same allele or 2 of the same gene or factor. Okay, in Mendel's time, he didn't know what genes were. He didn't know what meiosis was. He didn't really know much about DNA for anything. So he called these things factors, and he gave them letters. Okay, we know them to be the actual genes or versions of genes from the mom and the dad. So if we look at the F1 plants, so this is when they mated the F1 plant by itself or by used its own genotype and mated them together. Okay, then these ones each have a chance of giving a big T or a little t to their baby plants. So what happened is if this one gives a big T and this one gives a big T, we end up with a big T plant, which would be tall. If the one on the left gives a big T and the one on the right gives a little t, we end up with big T, little t, still tall, okay, because it has a dominant allele. If the one on the left gives its little t and the one on the right gives its big T, we still end up with a big T, little t, so another tall plant. So that's 3 tall. The last possible combination would be little t on the left and a little t on the right to give little t, which gives us the short plant. Okay, so in the monohybrid cross, he found a ratio of 3 to 1. Now, that ratio is what they actually see. Okay, we saw 3 tall and 1 short plant. So when we talk about what we actually see as the trait, that is called the phenotypic ratio. The phenotype is, pheno is for physical appearance. Okay, so what you actually see or observe. So they saw 3 tall plants to 1 short plant. And that's how you write a Mendelian ratio. So if they ask you to write the right ratio, the phenotypic ratio or the genotypic ratio, then we usually write it with a colon. The genotypic ratio shows the gene possibilities. So if you look at the gene possibilities, that has to do with the letters, the possible genes that were combined to get the offspring. So if you look at them, there's one big T. Okay, so here's the one big T. There's two big T little t's, two big T little t, to one little t. Okay, and usually you write it as um, the, the homozygous dominant, so that's the two big ones, then the heterozygous, which should be one of each, and then the homozygous recessive, which is little t. Okay, so this was the kind of ratio he found every time he looked at a single trait, and he had 7 different traits that he looked at, right? So flower color, purple versus white, position of the flower, axial versus terminal, um, the shape of the pod, so smooth versus wrinkled. All of those times that he did all those over the 8 years, he found that this was the kind of results he got. Okay, so as long as we're talking complete dominance, then this was the type of ratio that was found. Okay, then this is showing the same kind of thing, but for the round versus wrinkled. So this is the parent generation. Okay, so the P generation, right? Where you have the pure breeding round versus pure breeding wrinkled. So this is the dominant trait because we can look if all the babies were round, that meant that it's the dominant trait. So it has big R, and usually we use the letter to represent whichever trait was dominant. So we wouldn't use W because W would start wrinkled, and wrinkled is the recessive. Okay, so we use R for round. So big R, because it's pure, meaning homozygous for big R. And this parent was pure for wrinkles, so little r. Okay, in the F1 generation, they found that all of them were round, so that meant that they were heterozygous round. They'd get a big R from the round parent and a little r from the wrinkled parent. Okay, so when he self-pollinated them, it meant that he took the pollen with the big R, little r, and he made it with the ovules that were big R, little r. Okay, so again, this parent on the left could give a big R, and the one on the right can give a big R to give big R, big R. The one on the left can give a big R, and the one on the right can give little r to give big R, little r. The one on the left can give little r, the one on the right can give its big R, and that gives us another big R, little r. And then the last possible combination would be a little r and a little r to give us the one wrinkled. Okay, so again, a phenotypic ratio would be 3 round to 1 wrinkled. And if you were looking at the genotypic, that's to do with the letters, you'd have 1 big R, big R. Ooh, let's see if I can, why am I not using this? This would be a lot easier. Trying to use the mouse. Big R, to 2 big R, little r, to 1 little r, little r. Okay, so same ratio each time. And I think that was the, no, okay. So, summary of what happened in terms of um, what they thought might happen. Originally, they thought if you combined well, let's see, if you combined a tall plant and a short plant, you'd think that maybe you'd get plants that were halfway between. But instead he found that they were all the babies were tall. So that meant that the short factor was hidden. Okay, so he said that the tall one must be dominant and the short one must be recessive. So he formulated the principle of dominance. Okay, so any of these laws you should be able to recognize. Okay, maybe for match or multiple choice or whatever, explain a little bit. Okay, so principle of dominance: when individuals with contrasting traits are crossed, the offspring will express only the dominant trait. And I included in here that's if we're talking complete dominance, because later on we learn about other types of inheritance patterns, okay, more complex ones than he understood in those days, because once we understood about meiosis and possible crossing over, things like that, then things can be a little trickier. Okay, some inheritance patterns are different for different traits. All right, so in the F2 generation, he saw, so in the F1s, he said they were all showing the dominant trait. In the F2, we found there was that 3-to-1 ratio, or 75. Sometimes we talk in terms of ratios where we have the colon. So when we talk Mendelian ratio, usually writes it with the colon, but we can also talk in terms of percent. So a 3-to-1 ratio would be the same as 75% to 25%, right? So he said each parent in the F1 generation starts with 2 hereditary factors, 1 dominant and 1 recessive. Okay, so if we were talking tall, then this is what they meant. Okay, each parent, when we talk about a cross in the F1s, was heterozygous. Okay, they have 1 of each type. That time, he talked of them as being hereditary factors or units, but we know them to be the alleles or different versions of the genes. The factors separate in the parent. Only 1 from each parent is contributed to the offspring. Each offspring inherits 1 factor from each parent. If the dominant factor is present, it will be expressed even if the recessive one is present. Okay, so what we see is the dominant trait. So this gives rise to the first law of heredity, the law of segregation, which said inherited traits are determined by pairs of factors, which we know now to be genes or alleles of the genes. These segregate or separate in the gametes with 1 in each gamete. Okay, so we've already done this and talked about this part. Okay, today we know that the factors he spoke of, or units, were really genes. And genes can occur in alternate forms called alleles. So, for instance, this is showing the mother and father. So we'll say, let's say this is the female, or from the ovule, and this is from the pollen. Okay, so mother and father chromosomes. Okay, and any gene along the chromosome has a specific location. Okay, and locus, see this word here is probably too small on your sheet, but locus means location. So every gene has a specific locus along the chromosome. This particular gene goes for flower color. Okay, so the locus or flower color gene, and it's in the exact same spot on the chromosomes. So it's coding for the exact same trait, flower color, but it can have a slight different sequence in the DNA, which means that it could cause them to have a different version of that particular trait. Okay, so this would be the allele or version of the gene that codes for purple flower. And this one down here would be the allele or version of the gene, same gene, okay, but the version of the gene that codes for white. So since purple is dominant, we would say that's the P allele, and white is recessive. We'd say this is the little p allele. Okay, so alleles are um, different versions of the gene. Okay, so when we talk in terms of dominant and recessive traits, we use we already kind of talked about this a little bit, but anyhow, for the dominant trait, we use the uppercase letter. Recessive trait, we use the lowercase letter. And for the dominant trait, we usually use a letter that represents that trait. So if it's tall, tall is the dominant one, we'd use a T, and we use big T. So um, homozygous, we said already, when two alleles are the same. Heterozygous, when you have one of each alleles, they're different. Okay, so purebred tall would be big T, big T. Purebred wrinkled would be little r, little r. Okay, because wrinkled is a recessive trait. So we'd use little r, little r. Okay, so these are just to kind of get you used to doing the letters. If purebred tall is big T, then purebred short would be the recessive trait, so it would have to be little t, little t. Heterozygous tall would be big T, little t. Heterozygous means one of each letter. If we were talking round and wrinkled, then purebred round would be the homozygous dominant, so it'd be big R, and heterozygous round would be big R, little r, because it has one of each. Okay, I think I'm going to stop this video right now because it's pretty long already. Ooh, yeah.